This video is reverse transcription. Reverse transcription is most often done by viruses in the Retroviridae family, most famously the HIV virus, and is basically exactly what it sounds like. The virus reverses the directionality of normal transcription and uses that to synthesize itself a complementary double-stranded DNA molecule called cDNA, from the original single-stranded RNA molecule, which is then inserted into the host cell's genome inside the nucleus. This process requires several other components as well, including specialized enzymes like reverse transcriptase and integrase, and the dNTPs floating loose in the cell's cytoplasm. Reverse transcription is so called because it synthesizes DNA from RNA instead of the other way around, synthesizing RNA from DNA, which is the normal direction for transcription. For this video, I'm going to go ahead and use retroviruses as my example, specifically the HIV virus, since it's the most well-known. After the HIV virus has fused to the host cell, and its capsid coating has dissolved, the single-stranded RNA, also called ssRNA, is released into the host cell's cytoplasm. And inside the capsid with the RNA is an enzyme called reverse transcriptase, which will bind to the 5' end of the RNA and use the dNTPs, which are essentially the loose nucleotides synthesized in the cytoplasm and left there to float, to form a complementary DNA strand called first-strand cDNA, which is what is in this second image here. After that step is completed, an enzyme called ribonuclease H, which is in blue, comes along and degrades the original viral DNA strand, leaving behind only the single-stranded complementary DNA. This DNA then undergoes polymerization, which results in a double-stranded DNA copy of the original viral RNA information called a viral DNA intermediate, or vDNA. Then integrase enzymes, which also come into the cell with the other viral components in the capsid, will clip off the 3' ends of both strands, and then they will sort of hook them together and carry them through a nuclear pore into the nucleus, where the integrase will then make a little tiny nick in the existing DNA, which allows the viral DNA to insert itself into the host cell's genome. As you can see in this image, it just pops in there, and where it is then replicated by the cell's own ribosomes as it goes through regular transcription, which can't tell the difference between the native and the foreign DNA once it's been integrated. Now, retroviruses aren't the only ones that use reverse transcriptase. Eukaryotic cells use reverse transcription to insert DNA copies into other parts of the genome in a process called retrotransposition, and telomeres use a specific reverse transcriptase to extend and maintain the ends of the chromosomes, among others, but retroviruses are the most well-known for using these enzymes. Because of their unique characteristics, the ability to synthesize DNA backwards, basically, reverse transcriptases are very useful in laboratory settings. They can convert RNA into DNA for things like RNA sequencing or molecular cloning and genomic analysis. Their most common laboratory use, however, is in reverse transcription polymerase chain reaction, which is shortened to RT-PCR. Now, RT-PCR can be used to amplify very small amounts of genetic material to allow for the detection of RNA at very low levels of expression, which makes it much easier to study in the laboratory setting than it would be if you couldn't do that. The ways, actually, the inhibition of reverse transcription is one of the main ways of treating viral conditions like HIV that are caused by that family of retroviruses also.